Good morning. I'd like to thank our two session chairs, Baumier and Panagiotis, for this wonderful opportunity to share some of our work with you. So I'll be talking today about molecular elucidation and engineering of stem cell fate decisions. And in particular, of course, there's very strong interest in the field to understand the molecular mechanisms that underlie the opportunity of a stem cell to either undergo self-renewal and amplify itself in number, or undergo differentiation into a variety of terminal cell fates. And basic knowledge of this can both enhance our understanding of the ways in which stem cells contribute to development as well as adult tissue function, as well as enable us to find paths towards harnessing these cells as cell replacement therapies. So as we well know, there's a variety of different cues that guide the differentiation and as well as self-renewal decisions of a stem cell. And the usual suspects include small molecules, growth factors and morphogens, as well as the solid phase, matrix proteins and proteoglycans. And in addition, we also know that these biochemical cues are modulated biophysically in a variety of ways to change the way in which they're presented to cells, and this can impact the way that the cells interpret these signals. So there's mechanical and topographical information. In other words, the matrix can be organized. In addition, there can be variation spatially. And finally, the focus of my talk today is going to be on temporal variation. That is, do the temporal features of an input signal alter the way in which the cell interprets that information? So there are a variety of motivations for studying this problem, and that's the fact that at some base level, stem cells aren't simply sitting in some static environment. The environments around them are continuously varying. So if you take the nervous system, which is really going to be my model system of choice today, we have electrophysiological variation on the time course of microseconds to milliseconds. There's a neurotransmitter release, and it's been shown that neurotransmitters can directly affect stem cells within neural tissue. At a faster rate, on the order of 1 hertz, there's a cyclic mechanical deformation due to blood pressure propagating from the bloodstream into the brain tissue. Moving on to hours or minutes to hours, we had changes in protein signals present inside the brain. There's a 24-hour periodicity associated with the circadian rhythm. And moving on to a period of days to weeks to years, there's changes in the environment associated with organismal behavior, for example, mood. So these raise a number of different questions, such as do temporal and spatial, for that matter, variations in signaling inputs regulate or impact cell fate? And how do stem cells, if so, respond to these signals? Do they simply sit there with a clicker and count the number of signals they receive until they hit some threshold level within some time window and then execute a fate decision accordingly? What if cells are receiving several different signals simultaneously? How do they integrate that information? And finally, if two signals are actually mutually exclusive in their influence on a stem cell, such as one signal telling the cell to self-renew and stay immature, the other one telling it to differentiate, how does a cell dynamically arbitrate between these two conflicting signals? So these are questions that I've been interested in for a number of years, but addressing them had to wait until we developed some technologies that enable us to very precisely control the input signals into a stem cell, and as a result, enable us to investigate how temporal variations in those inputs impacted cell behavior. So a number of years ago, on the order of 8 years ago, we began to work with a technology that's well known within the neuroscience field, but in a different way, and that is optogenetics. So optogenetics is a field that utilizes light-responsive proteins to dynamically control cellular signaling. So you're likely familiar with the fact that there are a number of light-responsive ion channels that either open or close in response to light, and these can be harnessed in order to perturb or control signaling electrophysiologically within a neuron. And this has been used in a wide way within the central nervous system to investigate how electrophysiological signaling controls cell function and even organismal behavior. However, we began to use it in a different way. And I wanted to first lay down the motivation for this, which is that light activation affords a great deal of control over an input signal. There's a high degree of spatial temporal resolution, spatial down to the diffraction limit, temporal up to the speed of light. It's very easy to control the intensity or the dosage of an input signal in the form of changing the intensity on an LED or a laser. And finally, most cells inside the body, with some exceptions like the eye, don't naturally have the ability to sense light. And as a result, if you take a signaling pathway inside of a cell and place it under the control of light, this is an input signal that's orthogonal to the majority of signaling inside of a cell. It doesn't affect it. So the concept would be to use a light switch not to control electrophysiology in this case, but to control a signaling pathway that regulates stem cell fate decisions. And we'd like this system to be modular in principle. So in other words, we'd like to be able to illuminate a cell and zap or activate a particular node within a signaling pathway, and then allow the impact of that perturbation, that activation, to propagate through the cell, and then measure downstream cell behavior. We'd like to do this in a way where we have very precise temporal control, so the on and off kinetics of that optogenetic protein would be relatively quick, at least on the time scale of a stem cell making a decision. We'd like it to be ideally modular so that we can use that same switch and engineer it in different ways to activate different signaling pathways. And finally, although I'm not going to be talking about this much today, we'd like to have spatial control over signaling pathways as well. So as I mentioned to you, this was first developed with light responsive ion channels, but we began to work with a different light responsive protein called cryptochrome 2, shown here as a crystal structure of Cry1. It's a close neighbor from Arabidopsis, a model plant organism used in botany studies. Plants, of course, are very light responsive, so it's possible to borrow proteins from them and copy and paste them into mammalian systems in an orthogonal way to potentially gain control over particular signals inside of a mammalian cell. So a number of years ago, we began to work with cryptochrome 2, fused it to a fluorescent protein, M-cherry, overexpressed it in just a HEC293 cell, and noticed interesting behavior, which is that once you illuminate the cell with blue light, the wavelength that cryptochrome 2 responds to, there's a very rapid oligomerization of this protein. And when you end up flipping off the light switch, the cells, the oligomers will actually dissociate and disperse back down to monomers. This process had a very rapid on kinetics on the order of 30 seconds until you ended up with a half maximal number of clusters inside of a cell. And the off kinetics were also relatively quickly. This turned off at a time scale on the order of 5.4 minutes. And this is rapid enough, for example, to potentially study signaling pathways that play a role in fate decisions. And I'll also mention that this process was very, very reversible, and you could go through it many different times. So here's the idea. There are many signaling pathways inside of a cell where oligomerization of a protein naturally plays a role in the activation of that signaling. So the concept is to take one of those proteins, fuse it to cryptochrome 2, then you could hit the cell with photons. The cryptochrome 2 will cause the oligomerization of that particular protein, and the cell doesn't know the difference of why that protein oligomerized, whether it was due to natural activation of the pathway or light-induced activation. But in either case, this protein will then become activated and begin to activate downstream signaling inside the cell. So we utilize this to place a number of different signaling pathways under light control for the first time in the field, including receptor tyrosine kinases, Rho GTPases, integrins, as well as Wnt. And today I'm going to be focusing in on a couple of signaling pathways, in particular Wnt. So shown here is the Wnt signaling pathway in its ground state, in its inactive state. And beta-catenin is a downstream transcriptional activator within this pathway that ordinarily becomes associated with the so-called destruction complex inside the cell, gets phosphorylated by a kinase called GSK3 beta, which ends up marking it for ubiquitination and proteasomal degradation. However, if there's an activation of an upstream canonical Wnt receptor, there are co-receptors, in particular frizzled and a protein called LRP56, then this ends up inhibiting the destruction complex, which enables beta-catenin to accumulate inside the cell, traffic inside the nucleus, and transcriptionally activate a number of downstream target genes that play roles in stem cell biology, developmental biology, cancer, et cetera. So Harold Varmus actually did the work that noticed or discovered that even in the absence of a Wnt ligand, clustering or oligomerization of the receptors is sufficient to be able to activate the signaling pathway. So receptor clustering plays an important role in the signal. So what we began to do was to take one of the Wnt co-receptors, LRP6, and in particular the cytoplasmic tail, fuse it to cryptochrome 2, and then hit the cell with light. This caused oligomerization, again, in a highly modular way. And this was reversible, which was important to us if we wanted to use it to study dynamics of activation of the pathway and downstream cell behavior. And again, the idea, the hypothesis is now that you have a cluster of these receptor tails, the cell doesn't know the difference for why, whether it's due to light or due to the addition of the natural ligand, for this receptor. And it should activate beta-catenin, and then beta-catenin would go into the nucleus, and we used a synthetic promoter reporter construct driving luciferase in response to beta-catenin's presence in order to quantify activation of that pathway. This is the result shown here, which is we show relative beta-catenin activity on the y-axis. These are now neural stem cells expressing our optogenetic Wnt switch, which we call opto-Wnt. And in the dark, we see there's very little activation of the pathway. And once we begin to activate the pathway at different periodicities, so this is a, I believe, 500 millisecond, half-second flash of light every 300 seconds for a period of time, every 60, every 30, every 10, every 5. So this is essentially a dose response curve. We find that there's dose-dependent activation of the beta-catenin pathway. And the ratio of the maximally activated pathway to the dark state is on the order of 200. So 200-fold induction or activation of this pathway in response to light. And this turned out to be even stronger than what the natural Wnt ligand, in this case Wnt3A, was capable of doing inside of these cells. So just to show you another proof of concept that you can control signaling both in time and in this case in space, this is a cell, just happens to be a fibroblast that is expressing an optogenetically activatable receptor tyrosine kinase. I'm not going to go into the details of that, but shown here in this heat map, the brighter the color, the more active the receptor tyrosine kinase within that region of the cell. And the other thing I'll mention is that the cell actually migrates naturally, chemotaxis in response to activation of that RTK. So we began to use a laser, a confocal microscope in FRAT mode, and using a laser, we're able to illuminate just a part of the cell. And you can see that there's local activation of the signaling pathway where there's illumination. And this is a second movie showing a LifeAct, which lights up actin filaments. And you can see that, again, laser here is able to induce the chemotaxis of the cell. So you can see the cell is essentially chasing the laser pointer. And once you end up changing the location to the opposite side of the cell does a 180, turns around, and responds and chemotaxis in the direction of the activation of the receptor tyrosine kinase on this side of the cell. So this just demonstrates there's a strong capacity to control signaling both in space and in time. Okay. The final piece of information I'll provide is in building this system is a light activatable board, which we call a lava board, light activation variable amplitude. And this is actually just to update this revision. This was published in Cell Reports a couple of months ago. So this is a board that we built from scratch, where each one of these circles here, and you can see this is customized for a 24-well plate. We also have 96 and 6-well plate versions of this, but each one of these circles contains underneath it an LED and a diffuser such that light is projected onto, in this case, every well of a 24-well plate that's placed on top of it. And these LEDs are controlled either with a wired signal, or we can do this wirelessly as well using an Arduino or a Raspberry Pi board or Pi system. And on the bottom, we have these heating systems, these heating pins, very chemical engineering, to be able to transfer heat out of the system and into the rest of the cell culture incubator, because this can be placed directly in an incubator to prevent the plate from getting overheated due to the electronics or due to the light. This is programmable, where we can change the light intensity of every single one of these wells independently, as well as the timing. And in work I'm not going to show today, it's possible also to print out masks so that you can actually pattern each one of these wells differently to get some spatial control. And this is just an example of patterning a field that hopefully is familiar to all of you on a single well inside of a, in this case, it was a 24-well plate. Okay, so now all we need is our stem cell system. And in this case, we're working with neural stem cells, as I mentioned to you earlier, and in particular, these are adult neural stem cells that were discovered to exist in the hippocampal region of the brain and to continuously divide and create brand new neurons on a daily basis on the order of hundreds of neurons in each one of our brains every day in a manner that modulates our ability to learn and remember, and also plays a role in degenerative disease. So there's a series of states that these cells transition through. There's a so-called type 1 neural stem cell, which has the capacity to differentiate into an astrocyte, or undergo progressive differentiation into a series of progenitor cell states that are still mitotic prior to terminally committing to becoming a newborn neuron. And importantly, others as well as ourselves a number of years ago discovered that these cells undergo differentiation into neurons in response to Wnt signaling. So it certainly wasn't an accident that we chose the Wnt signal to place under optogenetic control the first time that we did this work. So I'd like to go through a series of papers that demonstrate the concept that inside the brain, Wnt is not this static thing that's always present at 20 nanogram per mil concentration. It's actually dynamically varying. So it was shown a number of years ago that neuronal activity and activation of the transcription factor CREB stimulate the expression of Wnt in cultured hippocampal neurons. So the Wnt signal depends upon electrophysiological activity of neighboring neurons. In addition, it's been shown that more recently that neuronal activity actually decreases expression of a Wnt antagonist, a secreted frizzled protein. So again, neuronal activity is changing the signaling state, changing the signaling activity or levels of Wnt agonists and antagonists. It was also shown in that same work that running modulates Wnt signaling in the hippocampus, so exercise increases Wnt. There's work in another system, this is in the intestine, that there's a circadian rhythm periodicity of Wnt signaling. And finally, moving towards a very long time scale, Wnt expression progressively decreases with age in the hippocampal region of the brain, which actually parallels an age-associated decline in the ability of stem cells in the brain to generate new neurons. So there's certainly a lot of temporal variation in this input signal. So it's possible to isolate adult neural stem cells, to clone them, and show that a single clone of cells is capable of undergoing both self-renewal in response typically to an FGF signal, as well as capable of undergoing differentiation into the three major lineages of the adult brain, namely neurons, astrocytes, and oligodendrocytes. So we took these neural stem cells, overexpressed our opto-Wnt system inside of them, and in the dark, you can see that these cells are not expressing high levels of beta-tubulin, but once you flip on the light, over a period of 5 days, on the order of 60, 70, or 80% of these cells will undergo neuronal differentiation. So the culture conditions, the culture plate, the signals, the growth factor media that's present on both of these parallel cultures is identical. All that's different is that this well received blue photons, which activated the opto-Wnt system and induced differentiation. So we began to ask the question, does this cell respond simply to some average level of a signal that it experiences over a long period of time, or do temporal variations, do the details in the temporal way in which a signal is propagated or sent to a cell change the way in which the cell interprets and responds to that signal? So we put these cells on our lava board system, and then we had the capacity to deliver light with different temporal inputs. So you can see here, we're actually changing the frequency of a light signal, not the frequency of the wavelength, but the frequency with which we turn on and off the light switch. And we were able to stimulate the cells in a variety of different ways to investigate how temporal inputs impacted the way that they respond to signals. So shown here is a static, meaning that the concentration or the intensity of light wasn't changing in this experiment, a static experiment in which we simply did a dose response curve. So we figured out what the maximal dosage of light was that we could use before cells began to experience some light toxicity, because if you shine too high an intensity of blue light, eventually it will cause toxicity within the cells. We are way, way below that intensity. And we simply increased the intensity of light on these cells and saw a dose-dependent increase in the differentiation of cells into neurons up to around 60%. And this was exposure for 144 hours. We then began to change the input signal properties. So for example, we ended up changing the pulse. We initially pulsed the cells for different periods of time and then flipped off the light switch. And we saw that if you expose cells to different durations of an input signal, the longer you expose them. So if you expose them for 120 hours and then turn off the light for the last 24 hours till you hit 144 hours, then 60% of the cells underwent neuronal differentiation. So in other words, as long as the cells were in the light for 5 days, it didn't matter that they were in the dark for the last 24 hours. They're still going to undergo maximal differentiation. But you could take these two different axes and have a duration of an initial pulse, which was at maximal intensity. And so if the cells received that maximal intensity pulse for the first half of the experiment, that's the same amount of dosage as the cells receiving a 50% intensity for the entire experiment. So you could basically collapse these two axes and re-express it simply in terms of dosage rather than intensity or time. And if all that mattered was the amount of light that a cell received over that 7-day period, if it had a clicker and was simply integrating the amount of light signal it received, then these two curves should exactly overlap with each other. And they do not, which indicates that the temporal way in which the light is delivered matters to the cell. This is information that temporal variations will result in variations in cell response. We then performed a different kind of experiment where we ended up exposing cells to oscillations, where we simply flipped on and off the light switch with different periodicities, period of T represented here. So this is a stimulation cycle duration. It's simply a period of oscillation or illumination. And so even though we were flipping the light switch at different speeds within different wells, the light was on half the time. And so all of these cells received a 50% dosage over time. And if you move back here, you'll remember a 50% dosage corresponds to a differentiation of around 50% of cells. So if the temporal way in which the light was delivered didn't matter, then all the cells should fall in this line of 50% differentiation. However, we found that if we had rapid oscillations on-off light switches, then the cells behaved as if they were averaging this signal. They integrated the signal over time. But if we got slower and slower in the speed in which we turned on and off the light, we saw that fewer and fewer cells underwent differentiation. And in fact, these cells ended up executing a completely different fate decision, which is that they ended up undergoing apoptosis. So we find that you can expose the cells to the same dosage of a signal, but if you do it rapidly, the cells are able to average out that signal and to adopt one behavior, namely differentiation. But if you did it slowly, the cells couldn't integrate or average that signal, and they end up instead committing suicide. So we wanted to understand intercellularly the nature of the signal that the cells were receiving. So we took that same beta-catenin, that same Wnt responsive promoter, and placed it in front of, in this case, a fluorescent protein. And we exposed cells to a constant signal and then quantified how much fluorescence there was inside the cell. And under a constant signal, the fluorescence builds up, indicating that the cells are continuously receiving this Wnt signal. And if you do it with a relatively fast periodicity on the order of 12 hours here, which you can see corresponds to a cell that's undergoing differentiation well, within each peak of activation shown here with this blue stripe, the cells build up Wnt, and then they start slowly dying down between these pulses, but it doesn't die down all the way to zero, especially after the cells have had a few cycles to accumulate the signal. Whereas if we have a very slow oscillation, such as 36 hours, which corresponds to this cell culture where there's significant cell death, we see that there's a strong buildup of the signal, but there's enough time after you turn off the light for the signal to die down all the way to zero. So there are these windows of time where there's strong Wnt activation, but there's intervening windows of time where the Wnt signal is essentially zero, it's completely died down. So what does that do transcriptionally to a cell? So we ended up investigating using RNA-seq different patterns of activation of a number of downstream targets of this Wnt signal. And I'll mention as well that we observed this behavior, this death behavior, when we used light, when we used a Wnt ligand, and as well as when we used a CHR, a Wnt agonist small molecule pharmacological activator of the signaling pathway. We found in all of these cases that if you end up adding that signal for a period of time and then removing it, we see that the cells underwent death. So we used 3 different conditions. So light, the Wnt3, a natural ligand, as well as this pharmacological inhibitor of GSK3 beta, which serves as an activator of the pathway. And we either added the signal for the entire 48-hour experiment, or we added it for 24 hours and then removed it for 24 hours to simulate this periodicity, or at least one cycle of this periodicity where we added it and then removed it. And we saw a number of different patterns in which, in some cases, there was activation of a downstream transcriptional target, and then it stayed on, even though the signal was removed, but in other cases there was activation, and then it died out once the signal was removed. And we found in particular a number of these interesting ones where there was an initial activation, but then the transcript went away. And we found one in particular called GAD45, which serves as a cell cycle regulator. And importantly, it's been shown to serve as an inhibitor of cell cycle progression. And we hypothesized that inhibition of the cell cycle, of course, is a really important step for a cell to undergo terminal differentiation. And if you have a situation where there's an activation of this inhibitor of the cell cycle, but then it goes away, that this could potentially cause a cell to undergo some indecision or some tough signals that are potentially telling it to stop dividing, but then perhaps start dividing again. And we found in particular that if you end up taking cells, this is data you've already seen, or similar to data you've already seen shown in gray, where this is the percentage of cells that undergo neuronal differentiation, and wild type cells. And if you have constant signal versus addition of the signal for 24 hours and then removing it, you see that there's a significant drop in the number of cells that have undergone neuronal differentiation by the end of the experiment, several days later, if you have this pulsive signal. But if you end up overexpressing the GAD45, this ends up restoring the halting of the cell cycle and rescues neuronal differentiation to a good extent. And we see the converse behavior with apoptosis, that if you end up exposing cells to a signal for the entire period of time, that there's very little neuronal death or very little cell death. If you pulse, but then remove the signal, that there's a significant increase in cell death. But if you overexpress GAD45, it actually rescues cell apoptosis, it halts cell apoptosis. However, we see, and this is a rather busy slide, so I'm going to explain it qualitatively, we see interesting behavior in downstream targets that are associated with neuronal differentiation. So specifically, if you end up hitting a cell with a signal called CHR or with Wnt3a, there's an upregulation in this transcription factor in NeuroD1, which is important for differentiation into neurons. So if you overexpress this transcription factor, the cells will instantly turn into neurons. And if you pulse the cells with the signal, this NeuroD1 still goes up. So we have a situation where if you end up exposing cells to a Wnt signal for a long period of time, it overexpresses GAD45 to halt the cell cycle and simultaneously upregulates this transcription factor NeuroD1 to induce neuronal differentiation. But if you hit a cell with the signal for 24 hours and remove it, the transcription factor controlling neuronal differentiation stays on, but the cell cycle arrest goes away and the cells try to enter back into the cell cycle. So you have this conflicting signal of the cells trying to undergo differentiation while still trying to divide, and this ends up causing apoptosis and cell death. The last thing I'll note is that, interestingly, this behavior may play an important role in the adult brain because the vast majority of newborn cells in the adult brain, on the order of 80% of the cells that are born, never make it to being a neuron. They undergo partial neuronal differentiation, but they end up committing apoptosis. And so we hypothesized that this mechanism, this essentially signal proofreading mechanism, where the cells are exposed to a half-baked signal that doesn't enable them to fully undergo differentiation, this is a mechanism to remove that cell from the brain prior to having this half-baked, poorly differentiated neuron integrate into a neuronal circuit in a way that could aberrantly affect the behavior of this circuit within the brain. So I'd like to very quickly expose you to a different system that we've been utilizing, in which we've been utilizing OptoWinds, in which we've been utilizing this optogenetic activation of a key signal. And this is a signaling system that I showed you an example from adulthood, and this is a situation in which the signaling system plays an important role in development in early stage embryos. And one of the earliest fate decisions that a developing embryo has to make is to have cells to be committing to be in ectoderm, which will end up giving rise to the nervous system, versus mesoderm and endoderm, which give rise to the rest of the body. And this is a process known as gastrulation. And Wnt gradients are present within the developing organism during this time, in the developing embryo. And in particular, there is a migration of cells which are fated to become mesoderm and endoderm, where they migrate through the ectoderm and then begin to form this structure called the primitive streak. So there's a natural migration of one cell type away from, or two cell types, mesendoderm, away from the ectoderm. And Wnt plays a signal, or plays a role in this process, Wnt signaling. So we we took human embryonic stem cells and knocked into them the opto-Wnt system so we can investigate the role of Wnt signaling in this process. So these are our opto-Wnt cells. This was published in Cell Reports a little bit earlier this year. And we find that in the dark, none of these cells undergo differentiation, but we also have within that same cell a GFP reporter knocked into the brachyury locus. And brachyury is a gene that's expressed within cells that are undergoing mesendodermal commitment. And so if you flip the light on, we see that these cells begin to express brachyury, become GFP positive. And this occurs with a very, very high efficiency. 98% of these cells will turn on GFP and undergo mesendodermal differentiation. And it's also very dose responsive. So we began to investigate the role of Wnt signaling in this migration and patterning in the early embryo. And I'll just leave you with one teaser piece of data. This last data is on its way to being published. And what we did was we mixed together naive human embryonic stem cells that don't have the opto-Wnt together with cells that do have the opto-Wnt. And we began to investigate what would happen if we activated opto-Wnt by illuminating the full culture to simulate potentially the activation of Wnt signal inside the cells that are undergoing mesendodermal differentiation, as you see within this gradient. And these are the cells that are well mixed together. Cell tracker marks every single cell in the culture. M Venus, in this case, marks the cells that are the opto-Wnt. So you can see a fraction of these cells that are cell tracker positive are also opto-Wnt positive, but not all of them. And initially, these are very well dispersed. Once you end up hitting the light switch and turning on the signal, you see that cells divide over time, but there's also a segregation, that these cells will actually phase separate from one another over time, such that the M Venus, the opto-Wnt, the Wnt activated cells will exit the colonies that contain the other cells and will phase separate into different structures. And we're currently studying this behavior both in 2D as well as in 3D as a model for gastrulation, as a model for this structural formation and separation of cells at different fate within the early embryo. So with that, though, I would like to leave you with a couple of parting thoughts, which is that stem cells are very, very interesting model systems to study cell fate decisions that take place within both adult as well as embryonic tissue. However, these fate decisions are very dynamic in nature. The environment surrounding the cells dynamically present information to a stem cell. And as a result, to study the relationship between signaling dynamics and cell fate and structure formation within tissues, we need to have temporal control over these signaling pathways. And optogenetics is a very powerful tool that enables us to gain that control. So I'd like to close by acknowledging the people who actually did this work. The work on optogenetic or opto-Wnt in adult neural stem cells was the product of Alyssa Rosenblum and Lucas Bugaj. The work on the opto-Wnt within human embryonic stem cells was the product of Nicole Repina and Xiaoping Bao. So thank you very much for your time and attention.